Hi everybody and welcome to our module on translation. Shown on the screen is the summary slide from the video on transcription. In that video I talked about how a DNA strand like this one at the top of the screen could be transcribed into a piece of mRNA, and in the nucleus the mRNA would undergo several modifications. A cap would be added, a poly A tail would be added, all the introns would be removed, and once those modifications had occurred the mRNA molecule could move into the cytoplasm. In the cytoplasm, mRNA is translated into a protein, and that's the process we're going to discuss in this video. And I want to clear away my ink here just to make an important point. I've shown the DNA coding strand in this picture here. The coding strand has the 5' end on the left and the 3' end on the right. And I explained the difference between the coding strand and the template strand in the video on transcription. I've also showed the piece of mRNA from the 5' end on the left to the 3' end on the right. And the reason that DNA is usually shown as the coding strand and mRNA is shown going from 5' to 3' is because mRNA is read in the 5' to 3' direction. What that means is that whichever amino acid is coded for here at the 5' end will be the first amino acid added to a protein when the mRNA molecule is translated, and then the translation process will move in this direction from 5' to 3' to synthesize a protein. So translation is the process of synthesizing a protein using mRNA as a template. It occurs in the cytoplasm on ribosomes, and the way it works is that tRNA molecules bring in amino acids to the ribosomes for assembly into proteins. We're going to get into the details of translation in this video, but first I just want to give you the big picture. So shown on the screen is a molecule of messenger RNA. This is the 5' end over here on the left. This is the 3' end over here on the right. The piece of messenger RNA is bound to a ribosome, and then tRNA molecules are bringing in amino acids, and the tRNA molecules are binding to the mRNA, and the amino acids are being linked into a growing peptide chain. That's how our body makes proteins. Let's talk about ribosomes a little bit. Some ribosomes are found free in the cytoplasm. Others are bound to the endoplasmic reticulum. The bound ribosomes are what give some parts of the endoplasmic reticulum a rough appearance, and this section of the endoplasmic reticulum is called the rough endoplasmic reticulum. And I talk about this in more detail in the video on cell structure. Ribosomes contain ribosomal RNA and also proteins, and they are arranged as a large and small subunit that binds to messenger RNA molecules. And the size of ribosomes is measured in an unusual metric called a Svedberg unit. This is a measure of the rate of sedimentation by centrifugation, but the bottom line is the Svedberg unit is a measure of ribosomal size. Prokaryotes or bacteria have 70S ribosomes made up of two subunits, a small 30S subunit and a large 50S subunit. And this is confusing because 50 and 30 do not add up to 70, but that's just the way the Svedberg units work. To make it even more confusing, the small subunit has a 16S size piece of RNA plus proteins. The large subunit has a 5S piece of RNA, a 23S piece of RNA plus proteins. And it's pretty low yield to memorize the size of all these ribosomes in prokaryotes, but what you should know is that the sizes of ribosomes in prokaryotes are different from in human cells, and this means that protein synthesis inhibitor antibiotics are drugs that target ribosomes only of the size found in bacteria. So for example, aminoglycosides and some other antibiotics that I discuss in the infectious disease videos target components of bacterial ribosomes. They do not have effects on human cells because human cells have different sizes of the ribosomal units. So in eukaryotic or human cells, the ribosomes are 80S in size. They're made up of a small 40S subunit and a large 60S subunit. The small subunit has an 18S piece of RNA plus proteins. The large subunit has several pieces of RNA ranging in size plus proteins, and it's pretty low yield to memorize all these sizes. The key point to know is that bacteria have different size ribosomes compared with human cells. Now let's talk about tRNA. tRNA are RNA molecules that transfer amino acids to proteins. That's why they're called tRNA. They are synthesized from DNA by RNA polymerase III. And one thing you should know about tRNA is that these are very unusual RNA molecules. They have an unusual shape, which I'll show you in a minute. And in addition, many of the bases are chemically modified. They are not the normal bases you find in RNA. For example, on the bottom right side of the screen is the chemical structure of guanosine, which you can find in many RNA molecules. In tRNA, you often find N,N-dimethylguanosine. It looks like guanosine, but it has this structure added to the molecule. This is an unusual base, and this is the kind of thing you find in many tRNA molecules. tRNA has a very unusual clover leaf shape. This is a picture of what it looks like at the top right side of the screen. At the bottom of the screen is another picture of the clover leaf shape showing the nucleotides. The clover leaf shape is the secondary structure of tRNA. The primary structure of the molecule is the sequence of nucleotides. The secondary structure is how it folds into the clover leaf shape. The reason it forms the cloverleaf shape is because of base pairing within the molecule. If you look at this picture at the bottom right side of the screen, bases are pairing with each other within the cloverleaf molecule. This is very unusual. Usually bases pair with another molecule, such as DNA pairing with RNA, but in this case, tRNA is pairing with itself. tRNA is a short molecule. It's only about 70 to 90 nucleotides in length. This is very tiny relative to many other nucleic acids found in the body. And there are four key portions that you need to understand for the Step 1 exam. The first part is the anticodon loop at the bottom. The second part is the D loop, which is part of the D arm of the cloverleaf. Third part is the T loop, which is part of the T arm of the cloverleaf. And the final part is the 3' end. We'll talk about all of these in the next couple of slides. So let's talk about the anticodon portion of the cloverleaf first. The anticodon is 3 nucleotides on tRNA that pairs with complementary mRNA, and correct pairing brings in the correct amino acids to synthesize the correct protein. So this is shown at the bottom of the screen here. We have a GAU on a piece of mRNA that is complementary to CUA in the tRNA, and therefore this molecule of tRNA, which has CUA, will bind here and bring in the appropriate amino acid. And the anticodon portion of tRNA molecules is part of the basis for our genetic code. Shown on the screen are the different sequences of nucleic acids found in mRNA and which amino acid they code for. So for example, a UUU in mRNA codes for the amino acid phenylalanine, and the reason it codes for that is because the UUU codon in RNA is complementary to an anticodon in tRNA that is linked to phenylalanine and will bring in that amino acid. The D loop gets its name because it contains an unusual nucleotide called dihydrouridine. In the middle of the screen is the structure of uridine as you would normally find it in a molecule of RNA. The D loop contains molecules of dihydrouridine. It's called dihydrouridine because 2 hydrogens have been added to this double bond here to make it a single bond. And you can see in this picture on the right side of the screen that we've got 2 of these dihydrouridine nucleotides present in the D loop. And the D loop is important because this is the portion of DNA that's recognized by the enzyme aminoacyl tRNA synthetase, and we'll talk later about what this enzyme does. Now let's talk about the T loop. This portion of tRNA gets its name because it contains a sequence of RNA called the T-Cy-C sequence. The T-Cy-C stands for ribothiamidine, that's the T, the Cy is pseudouridine, and the C is cytidine. If you look at the bottom left side of the screen, this is the chemical structure of ribothiamidine. Thymidine is not normally found in RNA. It's normally found only in DNA. And thymidine normally exists as a deoxyribonucleic acid, meaning it is missing this hydroxyl group. This is a molecule of thymidine like you would find in DNA, but it has a hydroxyl group here, meaning it is a ribonucleic acid. This is a very unusual nucleotide, and it's found in the T loop of tRNA. The Cy in the T-Cy-C sequence stands for pseudouridine. This is the structure of normal uridine in the middle of the screen. Pseudouridine looks a lot like it, but the nitrogen is in the wrong position in the base. Therefore, it's called pseudouridine. And then the C is cytidine as you would normally find it in RNA. So if you look at this clover leaf at the top right side of the screen here, you can see the T-Cy-C sequence that is always found in the T loop. And this portion of the tRNA cloverleaf is necessary for tRNA to bind appropriately to ribosomes for translation. And finally, let's talk about the 3' portion of the tRNA molecule. The 3' end of a tRNA cloverleaf structure always ends in the nucleotide sequence CCA, as shown in this picture on the screen. And it is the hydroxyl or OH group of adenosine that is going to attach to the amino acid that tRNA is bringing into the ribosome and mRNA to synthesize a protein. So that's a nice segue into the topic of charging. Charging is the process of linking an amino acid to the 3' end of a tRNA molecule. Each tRNA molecule is linked to one amino acid, and the enzyme that adds the amino acid to tRNA is called aminoacyl tRNA synthetase. It adds amino acids to tRNA and it uses ATP to carry out this role. On this slide, I've depicted the process that aminoacyl tRNA synthetase catalyzes in order to charge a tRNA molecule with an amino acid. The process begins with an amino acid as shown here. All amino acids have a similar structure. They have an amine group, a carboxylic acid group, and a side chain. The side chain is different from one amino acid to another. As I mentioned on the last slide, this process requires ATP, and the first step is actually to add the amino acid to the ATP molecule. Two of the phosphates are lost and the amino acid is linked up to AMP. This forms an intermediate called an amino acid adenosine monophosphate. Next, the AMP molecule is split off and the amino acid is added to the end of the tRNA molecule, and this forms an aminoacyl group. Hence, the enzyme that catalyzes this process is called aminoacyl tRNA synthetase. In the human body, there are many different forms of the enzyme aminoacyl tRNA synthetase. Shown on the slide here are all different amino acids, and in general, there is one aminoacyl tRNA synthetase enzyme per amino acid for most eukaryotic cells. In other words, there is one enzyme to attach glycine to the correct tRNA, another enzyme to attach other amino acids to the correct tRNA. Now, many amino acids have similar structures, and if a tRNA molecule were to be mischarged with the wrong amino acid, that could lead to the wrong amino acid being inserted into a protein for a given mRNA codon. Luckily, this is avoided by a process called hydrolytic editing. So aminoacyl tRNA synthetase scrutinizes the amino acid that is going to be added to the end of a tRNA molecule. And if the amino acid is incorrect, then that amino acid can be hydrolyzed either from ANP or from tNRA. If you look down at the bottom of the screen, this is the AMP molecule that is bound to an amino acid. And so if the incorrect amino acid is bound, then aminoacyl tRNA can lop this off, and then that amino acid won't be added to tRNA. Aminoacyl tRNA can also lop off the amino acid if the wrong one is at the end of the tRNA molecule. And so through this process of hydrolytic editing, our cells in our body have increased accuracy of the charging of tRNA. So now we've talked about tRNA and we've talked about ribosomes. Now we're ready to talk about how the whole process comes together to synthesize a protein. So first, a general point. Amino acids have an N-terminal end and a C-terminal end. If you look at this drawing on the screen here, this is the N-terminal end with the amine group. This is the C-terminal end with the carboxylic acid group. And protein synthesis always occurs such that additional amino acids are added to this end here, the C-terminal end, and this begins to link up amino acids in a chain going in this direction. Protein synthesis is generally divided into three stages: initiation, elongation, and termination, and we'll go through each of these stages one at a time in the next few slides. Shown on the screen is a strand of mRNA with a 5' end at the left and a 3' end at the right. It has been bound by a ribosome indicated by this red structure here. There's a large subunit at the top and a small subunit at the bottom. And each of these little colored squares are meant to depict different nucleotides. So the first thing you should be aware of is that ribosomes have 4 binding sites. One is for the mRNA to bind. That's what's shown on the screen here. The 3 additional sites are called the A site, the P site, and the E site. Each of these sites is a codon in the mRNA strand that will be important for synthesizing a protein. The A site is the location of amino acid binding. This is the codon that is going to bind to the anticodon on a charged molecule of tRNA. The P site will hold the tRNA that is attached to the growing protein chain. I'll show you that in a minute. And then finally, the E site is the site for the exit of tRNA after it has given up its amino acid to the growing protein chain. Initiation of protein synthesis always begins with an AUG codon on mRNA. This codes for the amino acid methionine in eukaryotic cells, or for an unusual amino acid called N-formyl methionine, or FMET, in bacteria. At the bottom left side of the screen is the structure of the amino acid methionine. N-formyl methionine has an extra group added to the nitrogen, hence the name N-formyl methionine. The tRNA for either methionine in eukaryotes or FMET in bacteria binds directly to the P site to begin the process of protein synthesis. This amino acid is usually not part of the final protein, and it's usually removed later by protease enzymes. It's added only to get the process started. And one important point here is that FMET is not usually found in eukaryotic cells. Therefore, if there's lots of FMET hanging around the body, that's a trigger to the immune system that there may be bacteria in the body. So for this reason, we have evolved so that FMET triggers chemotaxis of neutrophils as part of the innate immune system. GTP is hydrolyzed as part of initiating protein synthesis, and in eukaryotic cells, initiation of protein synthesis requires additional proteins called initiation factors that help to assemble the ribosomes in the tRNA. At the completion of the initiation portion of protein synthesis, we have the tRNA for either methionine or FMET bound to the P site. We now enter the part of protein synthesis called elongation, where the protein chain will be elongated. Elongation is usually divided into a sequence of 4 steps, which I'll show you on the next few slides, and this process uses proteins called elongation factors. In bacteria, the elongation factors are called EF2 and EFG. In eukaryotic cells, they're called EF1 and EF2, and these elongation factors hydrolyze GTP to GDP as part of the process of elongation. And just an aside here, elongation factor 2 in eukaryotic cells is a target of bacterial toxins. I talk about this in the infectious disease section, but diphtheria toxin produced by Corynebacterium diphtheriae and exotoxin A produced by Pseudomonas both target elongation factor 2 and thus inhibit protein synthesis in eukaryotic cells. The first step in elongation is for a charged tRNA molecule to bind to the A site. If you look at the bottom of the screen, the A site and the P site are right next to one another. The P site holds the growing protein chain. Initially, the P site contains only methionine or FMET, but as this process goes on, it will hold a growing protein chain. So the first step in elongation is for a charged tRNA molecule to bring in the next amino acid and bind to the A site. The second step in elongation is for the amino acid in the A site to be joined to the growing protein chain. This linkage is catalyzed by the ribosome itself. This is very unusual. Usually enzymes are proteins, but in this case, the ribosome is acting as the enzyme. It's sometimes called a ribozyme. The ribosome has enzymatic activity called peptidyltransferase. This activity is found in the large ribosome made up of RNA, not proteins, and this catalyzes the linkage of the amino acid in the A site to the protein chain in the P site. Step 3 is for the ribosome to move down the mRNA towards the 3' end. This is called translocation. When this occurs, the protein is now moved to the P site and the A site is empty, and a piece of tRNA is now sitting in the E site ready to exit. And then step 4 is for the tRNA molecule in the E site to exit, and now we're ready for another tRNA molecule to come into the A site and bring in another amino acid. Translation terminates when an mRNA stop codon is encountered. There are 3 stop codons, UAA, UAG, and UGA. These codons in mRNA are not recognized by any tRNA molecule. In other words, there is no tRNA molecule with an anticodon to these 3 codons in mRNA. They do not specify an amino acid. When a stop codon is encountered, releasing factors will bind the ribosome at the stop codon. This will catalyze the addition of water to the protein chain, and that will create an OH group at this end of the molecule, and we now have synthesized a protein chain. After translation, many proteins undergo post-translational modifications. These are modifications to the protein molecule that are necessary to make it functional. They include things like folding the protein. They also often include addition of other molecules. I'll go through just a couple of examples of post-translational modifications. Phosphorylation is when an amino acid residue in a protein is phosphorylated, meaning a phosphate group is added. The enzymes that do this are often called kinase enzymes. They add phosphate groups to other structures. Glycosylation is the formation of sugar amino acid linkages. There are many types of linkages. An N-glycosylation is addition of a sugar to a nitrogen in a protein. O-glycosylation is addition of a sugar to an oxygen, and C-glycosylation is linkage to a carbon. And all of these linkages create glycoproteins. Hydroxylation is the addition of hydroxyl groups to proteins. This is a very important part of collagen synthesis, which I discuss in the video on connective tissue. In collagen synthesis, proline and lysine amino acids in proteins are hydroxylated. For example, proline is hydroxylated, meaning a hydroxyl group is added. This creates the amino acid hydroxyproline, which is found in collagen molecules. Methylation is the addition of a methyl group, a CH3 group, to a protein. Acetylation is the addition of an acetyl group, a CH3CO group, to a protein. This is what an acetyl group looks like shown on the screen here. And then finally, ubiquitination is the addition of ubiquitin, a small protein, to another protein. Ubiquitin tags proteins for destruction in cellular structures called proteasomes. And I talk more about proteasomes and ubiquitin in the video on cell structure. And the final topic I'll mention in this video is chaperones. Chaperones are proteins that facilitate folding of other proteins. They bind to other proteins and ensure proper folding and stability. And the classic example of a chaperone are the heat shock proteins. Heat shock proteins are a family of proteins also called stress proteins. They are constitutively expressed. In other words, they're always present in cells, but their levels are increased in the presence of heat, a pH shift, or hypoxia. That's why they're called stress proteins or heat shock proteins, and they stabilize other proteins and maintain the structure of other proteins. This helps cells to survive in environmental stress. If not for these proteins, mild environmental stress like heat or pH changes or low oxygen levels could cause proteins to fall apart. But these heat shock proteins, which function as chaperones, keep them stable so that cells can survive in unfavorable environments. And that concludes our video on translation.